Physics. So I've been thinking a lot this week about sort of the order in which we teach things. Normally, this would be the first lecture, and there would be a very standard progression of lectures during a week like this. But because it's the summer and people travel, I sort of had to fit people in where I could, and I'm not so sure that's a bad thing. I think sometimes it helps to do some labs and be thrown into knowledge and education, and then come back to the basics and really firm up your background knowledge once you have some idea of what in the world you're talking about. I think about this a lot related to medical school, where one of the first classes you take is anatomy, and you have absolutely no clue as to what the concept of all these, elaborate nerves you're carefully dissecting are. So with that being said, Paul's going to talk about some of the basic concepts that you've heard about, some, not all of them, but you've heard about some of these concepts already this week, but not really in sort of a structural and foundational way. So I think that today is going to be a very interesting lecture and very helpful to sort of pull some of your knowledge together. Paul.
Yeah, thanks. Yeah, and I apologize for having been away at the right time, but I agree with Becky. I think some of this stuff might resonate more now that you've seen a bunch of other stuff. So I want to go over only some of these things today. I want to dwell a little bit on really simple stuff. This actually also comes from a medical school lecture that the person who was speaking before me gave on just how big are things. How big are things? How fast do they move? If you have that bit of information in your intuition, it's surprisingly useful for other things. And that motivates a little bit of discussion of Brownian motion, which I assume you've already run into in somebody's talk. Yes? No thermal energy. Okay, even better.
Well, yeah, a little bit in Andrew's talk.
Have you seen? Okay, Anders must have talked about some of this, right? And but I do want to talk a little bit about Brownian motion as opposed to motor-driven motion. And in the context of that, I'll give you a very short reminder of thermodynamics and kinetics, which I imagine you've had more recently than I have. But there are a couple of salient features which are just good to keep in the back of your mind. And then hopefully there's time, and I want to talk a little bit about the forces that drive protein structure, nucleic acid structure, all kind of cell membrane, everything that is kind of biologically interesting is not driven by covalent interactions that are kind of drummed into our mind by understanding protein nucleic acid synthesis, and also the way that drugs work. Drugs work in a very specific way, and most of biology is one step behind, or evolved when things are one step behind it. So I want to talk a little bit about the weak covalent interactions that dominate biology in water-containing environments, and then talk a little bit about how mechanical things change this kind of chemistry. I leave in the end of the slide deck a bunch of kind of introductory things, just to remind you, if you've forgotten, what the structure of protein, amino acids, nucleic acids, sugars are, and how they're assembled into macromolecules. There's no sense in going over that, but the slides might be useful to you. Okay, all right, so let's start. All right, length scales. So we're not online, right? So I can walk around.
Yeah, you can walk around.
Okay, so it's bewilderingly useful to keep in mind something about how big stuff is, because it really does help you understand what kind of effects might be useful. So the smallest things that there are kind of, well, smallest things we care about now are sort of atoms, small atoms, metal ions, stuff like that, are kind of they're less than a nanometer big. They're a few hundred angstroms big, or they're angstrom size-ish. So, and when they get assembled into molecules, those molecules are sort of small molecule drugs, stuff like, sugars, amino acids. Those things are someplace in this sub-nanometer range. They can get big, so something like a buckyball is one nanometer. when you start assembling relatively small still, but getting bigger molecules like phospholipids or fatty acids, in the diet, or sugar molecules or something, those things start getting to be a little bit bigger than a nanometer. So one kind of useful thing to keep in mind if you ever work on lipid membranes, is that a phospholipid is about 2 nanometers. A single phospholipid is about 2 nanometers long. It's much skinnier, but it's about 2 nanometers long. And then by the time you get to proteins, even small proteins like albumin, actin, tubulin, that kind of stuff, those things are like several nanometers. But proteins can become really big. So as the molecular weight goes up, and especially as they form secondary structures that make them filamentous, things like myosin or fibrinogen, those things are hundreds of nanometers long. They still tend to be fairly skinny, like a few nanometers in diameter, but the filaments themselves could be very long. Okay, so that's about the limit of how big you can make a protein. All right, so but you could of course make aggregates of proteins that could be much, much bigger, polymers and various kinds of assemblies. And one of the simplest assemblies that's in some self-sustaining is a virus. So most viruses are about 100 nanometers big, including HIV, SARS, Ebola, everything. Everything seems to be, if it's spherical, it's 100 nanometers big. It's never I don't think it's ever a micron, but it can't be much smaller because it has to have enough ingredients to do its job. All right, and so keep that in mind, that viruses tend to be, at least the spherical ones tend to be like 100 nanometer. Things that are elongated can be microns long. So PF1 virus, so one of these viruses, a bacteriophage that's excreted by Pseudomonas bacteria during cystic fibrosis infections. That's a virus that could be 4 microns long, but still only about 1 nanometer in diameter. Okay, so that's how big a virus is. Bacteria are like a micron. And actually, I should say this curious thing, it's not obvious why viruses can't get bigger. They don't, they're very small. Same with bacteria. Bacteria all seem to be like a micron, plus or minus a factor of 2 or 3. There are few, if any, bacteria that are 10 microns in diameter. They're all 1. All right, so that's it for kind of pre-eukaryotic stuff. Eukaryotic cells are all bigger. So and one of the reasons that they're bigger is that they got to figure out how to do more complicated things, because early eukaryotes, which are probably fairly small, swallowed up bacteria to make mitochondria. So the mitochondrion, which used to be a bacteria, was also like micron-ish. So the cell that contains it has to be bigger. And the smallest kind of mini cells are things like red blood cells or platelets, both of which are curious because they don't have cell nuclei in them. And they don't have well, at least a red cell doesn't have cytoskeleton, but it's useful to keep in mind that a red blood cell is something like 5 microns in diameter. But bigger animal cells and plant cells as well are often an order of magnitude bigger. So something like a fibroblast, and even more so, something like an embryonic stem cell, that thing has a diameter of about 30 microns, more or less. And when it's spread on the surface, it could be enormously big. It could easily be 100 by 100 microns in size. But when it's in suspension, it's about that same size. Not so different from plant cells, but they span a big range. They could be anywhere from a few tens of microns in diameter to a few hundred microns in diameter. So adipocytes, fat cells, depending on the state of the fat and the age and so forth, those things can get really big, 100 microns easily. Okay, so that, but never bigger than that, I think, at least when they're just in suspension. And then there are things that are not exactly somatic cells doing some job in the body, but things like pollen grains from plants or human egg. Those are bigger than 100 microns. Those things you start to be able to see by your eye. And things like frog eggs, you can pick them up with tweezers, are almost a millimeter big. All right, so that's kind of a useful thing to keep in mind, and especially when it comes to thinking about the mechanisms that could move stuff around in the interior. Oh, and the one thing I forgot that's not on this thing is a eukaryotic cell nucleus is usually about this big. That's one of the reasons why a red cell can't have a nucleus. The cell that made the red cell had a nucleus, but the same as the cell that made the platelet, that had a nucleus, but the nucleus is bigger than these little cells that pop out of the precursor cell. So eukaryotic cell nuclei tend to be 10 micron-ish big. Right, and those are fairly well preserved limits. All right, now, how fast does stuff happen? This is taken from a textbook, and there's too much stuff on here to talk about, but I'll point out a couple of the fastest things and the slowest things that are kind of maybe certainly relevant to cell biology and maybe relevant to mechanobiology. So you probably remember from, if you ever did spectroscopy or something else, that the movement of electrons and the conformation change, bending and stretching of bonds in an atom or a big molecule, those are very fast. Those things like bond vibrations happen on a time scale somewhere between a femtosecond and less than a picosecond. Things like absorbing a photon and sending the photon to an excited state in a molecule, that's still fast, but it's slower. That's more kind of picosecond-like. And so that becomes relevant for some of the fastest things that happen in biological stuff, which is photosynthesis. So when a chloroplast in a plant sucks up a visible light photon, it excites the rhodopsin or whatever it has in about a picosecond. But then what happens once that excited state is created, once it relaxes to do whatever it's going to do, generate conformational changes, send out fluorescence, create ATP, that has an enormous range of times over which that decay can happen. So the photon getting exciting the molecule, that happens super fast. What happens downstream can be somewhere between picoseconds and even like microseconds. All right, and then other things like how fast do we see things? So in your eye, once you absorb a photon and you have to transmit the excited state of the molecule that absorbed to create a neural signal so that you can process what you saw. And so depending on the animal and how much light there is and so forth, that process of converting an excited photon into a nerve impulse takes somewhere between probably much slower than a nanosecond, but up to almost a millisecond. So it takes time to process the information that you get into something that you can interpret. So you can't really see anything that's flickering faster, than 1,000 frames per second, right? So it'd be pointless to show a movie at 1,000 frames per second in real time, because you wouldn't be able to tell the difference between that and 100 frames per second, because your eye just can't process the information. Obviously, you can slow it down so you can see it. All right, and then stuff that is like related to signal transduction, helix coil transformations in a cell, opening up ion channels in response to some signal. That happens in this kind of like microsecond time scale. Still pretty fast, but it requires a lot of big things to move. So you cannot stimulate a cell faster than probably tens of microseconds, right? The machinery just can't respond to it that fast. And one of the reasons, and then what the cell does downstream of that is, of course, much slower. Stuff like folding a protein. Once a protein comes out of once the polypeptide gets threaded out of the ribosome, it isn't a protein for a surprisingly long time. It could take anywhere between like a few milliseconds, I guess, for very fast, very small proteins to immediately kind of squiggle up into their right shape, to tens of seconds. So lots of proteins take, and same with DNA, it takes a very long time to duplicate DNA because there are thousands of steps involved. Each one of those is, takes some time. So things like protein synthesis, DNA synthesis, it's probably useful to think of them happening over the scale of many seconds to minutes. All right, okay, so far. All right, so, and what else to tell you? Yeah, things like turning a bacterium or a eukaryotic cilium or flagellum, that happens in sort of it'll rotate at frequencies like maybe 100 hertz. The same with motors. Molecular motors like myosin that moves, or kinesin, or the proteins that move DNA around in the nucleus. Those things also take a while. They'll take a step, and then they have to recharge, take another step, recharge, take another step. And the fastest of those, I think, can cycle at about 100 hertz, not a lot faster. And so that cycling time that it takes for the motor protein to do its thing, to make a motion, get a new ATP, hydrolyze it, dump the ADP and phosphate, that takes time. And so that puts a strict limit on how fast things can move by motors. All right. Okay, so now how fast does stuff move around? So the fastest motion, which we'll look at when we talk about Brownian motion, is, and probably one of the reasons that bacteria are as small as they are, is that it takes about 10, 20 microseconds for a protein to diffuse from one part of a bacterium to another. As we'll show in some detail, it takes minutes for that same thing to happen in an animal cell. Much too long for diffusion to be useful. For bacteria, just random thermal motion is really good. That happens faster than one step of DNA polymerase. So a little molecule can zip from one part of the bacterium to the other faster than the machinery that duplicates DNA can take one nucleic acid and attach it to a growing nucleic acid chain. Okay, so if you need to provide stuff like ATP or do nucleic acids or anything to the DNA synthesis machinery, you don't have to send it in any directed way. You just let random thermal motions kick the new ingredients where they need to go. All right, and then the limiting time for a protein motor to move also puts a limit on how fast it can move a cargo. So a motor like kinesin or something, it would take a second. The very fastest linear kind of motors in the cell, they would take about a second to move a micron. Diffusion takes 10 milliseconds to do it. Motors take 100 times more time. So they're not useful for small things, but we'll see that they're very useful for big things. All right, and the reason that they're useful for big things is that by the time you get to an animal cell, and you ask, and a HeLa cell is a fairly kind of average sized animal cell, human cell. How long does it take diffusion to get from one side of a eukaryotic cell to the other? Instead of taking 10 milliseconds, it takes 10 seconds or more than 10 seconds. So that is too slow, for example, to provide stuff to DNA polymerase if that's, you need to either compartmentalize or do something else. All right. Okay, and then much longer things to think about. How long do big molecules live? And there's a huge range over which molecules live. So fairly expensive to synthesize, but also short-lived macromolecules like RNA, like messenger RNA. Once it's made, which probably takes on the order of minutes, it's dead in about 20 minutes. It's chopped up again and dead in 20 minutes, unless it's somehow protected. So things are synthesized, and then some things are degraded really rapidly. RNA turns out to be degraded really rapidly. DNA doesn't get degraded for years or millennia. That's why you can sequence, mastodons. But there's a huge range over which things are degraded once they're made. And that's also true for proteins. And one thing that's useful to keep in mind, especially when you hear talks about mechanical memory or something like that, or passing things from mother cells to daughter cells, is that some proteins are degraded faster than the doubling time of the cell, and some proteins are degraded slower than the doubling time of the cell, much slower. So this is especially true for things that leave the cell, like collagen or elastin in a lot of your connective tissue. A collagen molecule, once it's synthesized in the tendon, will stay there probably the rest of your life. Whereas the same collagen molecule that's synthesized in your gum is degraded in a day. That's why your gum or your cheek heals so quickly and your tendon doesn't, right? There's no way to get these materials to turn over. All right, okay, and then the organism itself. A yeast, or I think a bacterium, well, yeah, the bacterium itself will divide. If it has enough food, it'll divide in like tens of minutes, really quickly. Things like yeast, which are eukaryotes, but still small single-celled objects, they will divide slower, but not a whole lot slower, like on the scale of an hour. But most eukaryotic cells divide way slower, like once a day, even in culture. But the cells in your body don't divide that fast. They divide much more slowly. So cardiac myocytes, I think, divide once every, well, depending on your age, they divide once every 10 years or something. Okay, all right, so far, all right, so those little limits are useful. And I'll get back to this in a second, but there's also, if you are fond of thermodynamics, there's this. How much energy is there in stuff? A really important thing to keep in mind is that there's thermal energy for free everywhere because we live at 37 degrees. And that temperature gives kinetic energy to all the molecules that are sitting around. And that energy, right? Energy is the same, work and chemical energy and the heat are all the same thing in some sense. So average thermal motion, just KT, is kind of a lot. It's usually given in some, useless number like 4 times 10 to the minus 21st joules, or 10 times, 10 to the minus 21st calories. Not very intuitive, but this is probably the most useful thing to keep in mind that we'll go over today. 1 unit of thermal energy is, because energy is work, you can convert, and work is force times distance. 1 unit of thermal energy is 4 piconewtons times 1 nanometer. So that's a hugely useful thing to keep in your mind. And we'll see why that is. And it's like, the 40th of an electron volt. Okay, useless. Anyway, so that's how much energy you get for free. The non-covalent bonds, which we might have time to talk about at the end, are only about 5 times stronger. You know, stuff like hydrogen bonds or hydrophobic interactions. They're a little bit stronger, but not hugely stronger. So they're always at the risk of being broken by the thermal energy that's shaking everything around. More stable things like a carbon-carbon bond are orders of magnitude stabler, right? Thermal energy is never going to break a carbon-carbon bond. You have to actually do real chemistry to do it. And things like the energy in a, so here's kind of an interesting thing to keep in mind. The energy that you get by hydrolyzing an ATP molecule is this much. It's like, I forget what that is, like 70 kilojoules. But what's useful to keep in mind is that the energy you get from hydrolyzing ATP is only something like 15 or 20 times as much as thermal energy you get for free. It's more, but it's not infinitely more. It's only like roughly 15 times more. And the energy that plants get when they suck in a photon of green light is way bigger. It's more not. It's almost 10 times as much energy to make one ATP. So photosynthesis is fantastically useful for producing energy, as are, you know, solar cells. All right, okay, so that's enough for this. If you remember nothing else, remember this little thing, and that this is only 10 times bigger than that, roughly. Okay, so what's next? Oh yeah, how stiff are things? So mostly here I have just animal cells from some review. So the very softest things, at least in our body, that are still kind of solids are things like mucus, which are, you know, the scale of viscoelasticity, they're like a few tens of Pascal. So hopefully somewhere along the line you've run into, you know what a Pascal is? Okay, perfect. And then very, very, very soft tissues like bone marrow, or one of the softest organs in the body is the brain. Those are roughly 1 kilopascal-ish, maybe a little bit less, but something like a kilopascal. And then as you go through different soft tissues, they get stiffer and stiffer, but nothing that we would call a soft tissue in the body is ever more than 100 kilopascal, unless it's diseased. And so probably as you've heard, you know, elsewhere, all kinds of diseases, fibrosis, cancer, you know, all sorts of things are associated with changes in the stiffness of a cell. So for example, what is this? I guess this is a kidney. So these little, the kidney or a lung, I guess it's a kidney. The objects inside there are designed to have a particular stiffness that's close-ish to 10 a kilopascal. If you become fibrotic and you start laying down more collagen, the kind of interface between the extracellular matrix and the cell can become 10 times stiffer, and then stuff won't work. The same with mucus. Mucus is designed to be soft because it has to get cleared by these cilia that are moving it up from your airway so that you can cough it out. And those motors have a limited amount of work they can do, right? Because they're limited by the thermodynamics of ATP. And the mucus that we have, any sort of mucus covered epithelium, is designed to be soft enough so that you can move a sheet of it by these flickering cilia. If they get too stiff, because you don't make enough mucus, you dehydrate it, you fill it with bacteria or, you know, cigarette smoke or something, then, or if it becomes too fluid, because you've inhaled something that degrades the mucus, then either the cilia move, but they're just flickering in a liquid, or they get stalled because the stuff they're moving is too stiff. And so this softness of the mucus is there for a reason. If it's too stiff, things don't work. If it's too soft, things don't work. Okay, and then But all of these soft animal things are orders of magnitude, many orders of magnitude, softer than bacteria or plants, right? They have cell walls. They don't have lipid bilayer membranes. So the cell walls are typically orders of magnitude stiffer. They are even stiffer than things like cartilage, and they're almost as stiff as stuff like bone. Okay. All right, and that is relevant because there is also a wide range and a fairly tight limit of how much force organisms can produce. Here's a kind of a little episodic look at the kind of forces that are generated in the body. So the weakest forces, I think, that are generated in animals during development or normal homeostasis are the shear forces that are produced by fluid flow, which seems funny because the fluid is flowing like crazy down your arteries. But the hydrodynamics of fluid flow and the size of the vessel that they're flowing in tells you that the fluid shear stress that a cell feels on its surface is somewhere from a few tens of millipascal to maybe a pascal. It's fairly soft. So if a cell is going to respond to this fluid shear stress, it has to have a very delicate mechanism to sense very small forces. Bigger forces, like the forces that are produced by a growing tumor, or when a cell is pushing its way forward, or when a white blood cell is pushing its way through the endothelium out of the blood vessel, those things are hundreds of Pascal, reaching kind of a kilopascal. And a lot of things that have to do with cell protrusion also they develop protrusive stresses on the order of kilopascal. Why kilopascal? Because that's about how soft the cell is. Right? The force that is going to deform the cell has to be on the order of the elastic modulus of the cell in order to, or the object that it's trying to deform in order to have a significant deformation. So the kind of forces that animal cells develop would never deform a bacterium, because a bacterium is 10,000 times stiffer. So bacteria have to have a different mechanism. All right. But bacteria do develop all kinds of interesting forces, not by motors necessarily, but by things like osmotic pressure. So they can easily, within a biofilm, produce many kilopascals of stress by spitting in or taking up or spitting out ions or activating water channels or something like that. And they can construct kind of formations within something like a bacterial biofilm that can develop forces that are as big as the force developed by a muscle. So muscles can contract with like hundreds of kilopascal of stress. The reason that the reason that a organized muscle can develop so much more force than a single cell is because they got organized to work in parallel. And the reason that the muscle needs to generate so much stress is because it's not deforming a single cell, it's lifting weights and doing other stuff. But it's still using exactly the same mechanism that cells use to move themselves. And then there are other things that are specific for bacteria and fungi that have a cell wall. Because those cell walls are so enormously stiff, if you're going to deform them, you have to develop a whole bigger amount of stress. And the mechanism to develop those stresses is, I think, always osmotic. It's never by motors. It's always osmotic because osmotic pressure can easily develop megapascal of stress and deform things that are much too stiff for motors. Okay, so let's see.
I have a question.
Yes.
So is there a relationship between stresses that are, or like forces that can be generated by living organisms and how much force they can experience? Cause like I can push a car.
Yeah.
Like, or I can't push a car, that's my maximum, but if I get hit by a car, it's over.
Oh, yeah, there is a relation between, yes, for sure. There is a relation between how stiff a cell or an organism is and the kind of force that it has evolved to deal with. I think that's, I think that's really right. And you see this a lot when the forces that a cell or a tissue is subjected to. When that force changes, the cells in the tissue change too. That's the basis of why exercise will grow bigger muscles, and why your bones start atrophying, when people are weightless, right? Bones are also extremely sensitive to the forces that they feel. If they don't feel a force, they'll stop growing. If they do feel a force, then something kicks in that allows them to grow faster. And I think probably also this is why, I'm not exactly sure this is true, but I think this is why bacteria and fungi have really stiff cell walls, because they don't care if they get hit by a car. But they probably are subjected to all kinds of forces in nature that they need to be able to survive, because they don't have mechanisms of escape or anything else. But that's kind of a guess. The other thing that the stiffness of or the kind of forces that are generated is linked to are the kind of stresses that cells can detect and respond to. So the kinds of stresses or that our sensory apparatus is designed to respond to is also enormously different. So, for example, I think the very softest mechanical thing that we can sense is things like sound waves. So that's both true in plants and in animals. These very long objects in your hair cells can be bent by micropascals of stress. Stuff that would never deform bacterium, for example. And the same thing with fluid shear stress. These fluid shear stresses are, as I mentioned before, are in this kind of millipascal to Pascal range. So many objects in the body are too stiff to be deformed by this kind of thing. So cells that are supposed to respond to fluid shear stress have developed all kinds of long extended soft protrusions like the glycocalyx or primary cilium to sit in these fluid fields and feel the stress. Bacteria do it too. Bacteria grow these pili, these long protrusions that can feel fluid flow. And then bigger things like things that respond only to kilopascal stresses, those things are like the adhesion sites of proteins to the extracellular matrix. If you pulled on them with a millipascal, nothing would happen. You have to pull on them with a kilopascal or hundreds of pascals of stress, and then proteins will turn over, integrins will get activated, all that kind of stuff happens. And then there are other things that require bigger stresses, like that cannot any longer be generated by motors. You need things like osmotic pressure or the force of gravity on your bones to do something like stimulate bone growth or to open up ion channels in membranes, and even more so when these things have to happen in plants or animals. All right. Okay, that's it for these scale things, and I'm almost done with this. Actually, I'm going to skip over the thermodynamics because I want to tell you something more interesting. All right, Brownian motion. Thermodynamics is just, there's thermodynamics and there's kinetics, and they're both important. So if you've forgotten that, just read over the slides. But here's why a lot of what I told you before is relevant to mechano kind of stuff. All right, Brownian motion, where did it come from? Brownian motion was discovered by some botanist who had a microscope, and he put pollen grains. How big is a pollen grain? Oh man, it's like it's bigger than 10 microns. It's like less than 100 microns, about 10 microns, somewhere in that range. It's not an angstrom, it's not a foot, and that's really important, right? It's in this kind of 10 micron-ish thing. 10 to 100 microns. The reason that's important is because that is big enough for a microscope in the 1820s to be able to see, but it is small enough for thermal energy to move it. Right? And that's what this guy's discovery was, that he put pollen grains in water and saw that they moved around and didn't know why they moved around. They didn't know if they thought they weren't living objects because they had been desiccated for a zillion years. But they didn't know if they had a life force in them or not, but they were moving in a random way. He didn't think that it was a life force. And then whatever this is, 50 years later, a guy in France had the insight that the reason these little guys were moving was because of thermal motion of the environment. They still didn't know the molecular structure of water. They didn't really know anything or how to calculate anything, but that's what this guy's insight. The first real explanation was, or one of the first real explanations was from Einstein in one of his famous 1905 papers, and who had the realization that you could calculate how much energy is an object of the size of a pollen grain would be bombarded by the moving water molecules at room temperature. And he figured out a way that you could compute how fast these things would move if you knew two things. You knew, I just told you that. That all you had to know is how much thermal energy there is and how much resistance to movement there is. The resistance to movement's the viscosity, the thermal energy is 4 piconewtons times nanometer. All right, and okay, so this is the source of Brownian motion, and that's what caused this pollen grain to move. If the pollen grain had been a millimeter, you wouldn't see it move. I believe if you put a frog egg, which is almost a millimeter big, in water, you wouldn't see Brownian motion, it's too big. Because the 4 piconewton, 1 nanometer kick that you get for free isn't enough to really move it until you can see it. All right. Okay, so the reason that this thing is such a useful thing is because many molecular motors, when they take a motor step, they're generating about a piconewton, somewhere between 1 and 10 piconewtons of force every time the motor moves. But the reason they're better than thermal energy is that they can move farther, and they could also move unidirectionally. Thermal energy doesn't have a direction, it just goes everywhere. So you can't get it to go from here to there directly. It'll just get there randomly by fluctuating. But motors can get you there directly, and they can take a step that's bigger than a nanometer. Okay. So I'm going to so you can calculate how long it would take diffusion to move someplace, and you can calculate how long it will take a motor to move someplace. So let's look at the diffusion of a small particle or a big particle through something like the viscosity of water, and just ask how far does it take to get somewhere. So let's say here's the viscosity, it's the viscosity of water, and let's look at how fast something that's 1 nanometer big, like a molecule, or a very, very small protein, or a centimeter big. How fast does it go? And we want the velocity of the particle to be, approximately a micron a second. Okay, so let's see. Sorry. I have a typo here. Okay, let's just stay here. Okay, let's just stay here. Okay, so these are the things we're going to be interested in. We're going to look at how fast stuff moves by diffusion. We're going to look at how fast stuff moves by motors. And we're going to first do it for a small particle or any particle that has, diameter L and that's moving through something like that has a viscosity eta, and it's just driven by thermal energy. Okay, so how fast could we move something? If it's 1 nanometer, like on the size of a glucose molecule, and you plug in the relevant numbers there, that glucose molecule has a diffusion constant of 2 × 10⁻⁶ centimeters squared per second. Okay, so not super interesting, but that means to get from one side of a bacterium to another with this random diffusion time, it would take 1/100th of a second to do it, 10 milliseconds. Just by the algebra of plugging stuff in there. All right, so pretty good. So diffusion is, the diffusion constant is, distance moved squared divided by t. So if you solve for t, you get 1/100th of a second. That's why diffusion is so good for moving stuff around a small object. However, if the object is big, not super big, but big-ish, like a big protein, that's big protein's diffusion constant is 1,000 times smaller than this, just by plugging in to this equation. So if the diffusion constant is much slower, and then you calculate how long does it take to get from one end of the cell to another, there are 2 problems. One, the diffusion constant is slower to begin with because the object that you're trying to move is bigger. And the second thing is you want to move at a longer distance. So if you calculate how long does it take a protein to get from one side of a biggish cell to another, it would take 25,000 seconds for that same thing to happen. So you can't have diffusion work for a big cell, especially if you're trying to transport proteins, like a newly synthesized neurofilament protein to the end of an axon. That would take forever by diffusion. You have to do it somehow else. Otherwise, that thing would never evolve. All right, so I'm almost out of time. I want to tell you one quick thing. I want to tell you 2 quick things that you may have forgotten about. The non-covalent bonds that hold everything together, right? They are only a little bit stronger than thermal energy, and there's really 3 kinds that are very useful to think about. And they all have to do with the property of water. So if you just remember this, water has this almost unique liquid property that it is extremely polar, right? The oxygen part is slightly negative, the 2 hydrogen parts are slightly positive, and it's kind of V-shaped. It has a polarity in it. And so that turns out to be important because water molecules really like to coexist together. They want to be near each other. They want to exclude things that can't be part of their energetic interaction loop. Right, the reason that the water molecules want to get together is they want, right, the positively charged hydrogens to be near the negatively charged oxygen. That's what a hydrogen bond is. That hydrogen bond is only a little bit stronger than thermal energy, and it has a fairly short distance over which it's almost like a real bond, but not quite. Okay, and they're everywhere. Biology is just dominated with these things, and they drive the formation of all kinds of stuff. And the reason that they drive the formation of all kinds of stuff is that they surround any ion in the body or any charged object. Anything that has a real positive charge, like a magnesium ion or something, is immediately swarmed by water molecules. They don't actually make bonds with them, but they just surround it. If it's a negatively charged ion, the same thing happens. They swarm it. But now they swarm it by pointing their hydrogens to it rather than their their oxygens. And now what happens if you put in something that they don't like, that they don't have an attraction to, that's not charged? If you put into them something like a methyl group, of which obviously there are many, that will disturb this network of hydrogen, oxygen bonds that the water would like to put in. Every time you put in something that cannot participate in that, you disturb the energy profile of the thing that the water wants to make. And so that means that you have to pay an energy penalty to put something hydrophobic inside water. It can still happen, but you have to pay for it. And if you have two of these things together, you have to pay twice as much. But then something beautiful happens. If this thing can be next to that thing, then most of the surface area that the water doesn't like is now covered up. There are fewer water molecules that are unhappy energetically here than they are here. And so that's what this hydrophobic effect is. And it's enormously powerful and it's everywhere. And so it is the reason that we have cell membranes. If we weren't living in water, like pollen grains have membranes inside maybe, but the outside is not a lipid membrane. The reason we have lipid membranes is because we have this beautiful formation of phospholipids that are hydrophilic on one face and hydrophobic on the other side, right? And how long is this? 2 nanometers big. So the lipid membrane is about 4 nanometers thick. So very small and very fragile, but it is held together not because these molecules want to be next to each other. There are typically no attractive interactions in the lipid bilayer. They don't want to stick together. They don't care about each other. They're there only because they're excluded by the water. And that exclusion is a fantastically powerful thing. It drives, let's say, the formation of lipid membranes. It also drives the formation of the little lipid particles in which vaccines are now packaged and all that kind of thing. Okay, so I think I will leave it there because we're out of time. There's more stuff on like macromolecule structure at the end of the slides if you're interested in it. Okay. All right. Any other questions? Yes.
Just a sort of a general question, but so do you have any recommendations for resources for like going into the nitty gritty of like particle diffusion, like particularly between like porous substrates or I don't know, more complex geometries other than like, let's just say traveling across a.
Yeah, I'm trying to think whether there's a good review, like a good review article. Let me hunt around. I'll find something. I think there is something in a textbook I've seen recently that discusses this. And I'll find it and I'll post it. Is Annie still? I'll send it to you if you can just post it. But this issue of diffusion versus motor driven things has been around for a while, I guess, and partly it comes about because there are always arguments about why bacteria, or do bacteria have a cytoskeleton? Bacteria never seem to have anything that looks like myosin or kinesin. They might have things that look like actin filaments or something like that, but they don't have motors. They have motors that spin, that propel the whole bacterium, but they don't have anything inside. And that's one of the arguments about why they might not have evolved to have this, right? Evolution of motors is a eukaryotic thing. Yes.
So I'm working with a sort of ionic polymer, and when you're talking about charge, it made me think about how it interacts with water. In that case, would it be like because you said for like positive charge would be rotated one way and negative charge would be rotated the other.
Yeah.
Interacting with this ionic polymer, would it just be like each one of those molecules like rotate in different ways based on if it was near the,
It'd be each one of the waters.
yeah, like how would that, and I was just, when you were describing it, if it has both positive and negative, would that just change based on how close it is to?
Yeah, oh, that's a really good question. So I think what's not clear, and what people argue about quite a bit, is if you have something like a charge in the middle, which fairly strongly orients the positively charged hydrogens here, you induce a structure to this whole little cage that's not randomized. And in biology, this is true not just for like small point charges like metal ions. It's also true for long filaments like actin filaments and microtubules, and DNA are hugely negatively charged objects. So this whole line of charges also organizes a layer of water around that single long polymer. And it's not clear whether there is ordering of the next layer of water and the next layer of water on top of that. And it's, so that part I think is still a very super complicated, misunderstood or non-understood aspect of how cytoskeleton organizes the cytoplasmic space, because not only is there a kind of an organization of water around DNA, there's also an organization of magnesium ions or sodium ions around that filament. And whether the relevant thing is only the first molecules that actually in some sense touch the filament, or whether that ordering protrudes farther out, I think that's not clear. The other thing which is really important, I think, to keep in mind is that this kind of electrostatic and water organizing thing also makes it possible for one protein to bind really well to another protein and never actually contact an atom of that protein. So there are plenty of interactions. Tropomyosin, for example, the thing that lines the groove of the actin filament, I believe it's true that no atom of the tropomyosin polypeptide actually bonds with an atom of the actin filament. They all are interacting largely electrostatically through this kind of lubrication layer of water and counter ions that are in between them. In other cases, that's not true. In other cases, two proteins just slap together, especially if they have hydrophobic faces, then they are strongly driven to stack together. Did that?
Yeah, I guess the follow-up was just like specifically with the Switter ions, when like both positive and negative are present.
Yeah.
Like, because I'm just using a Switter ionic substrate, so I was wondering specifically how that might interact, but you still answered some of those questions.
So it's not, I think it's the interaction of the these so-called hydrogen bonds are still kind of like a regular bond. They happen because the electron cloud of the hydrogen and the electron cloud of whatever the negative thing is in the protein, those things are polarized towards each other, and they don't actually share the orbitals like you would in a covalent bond, but close. And so it is a real interaction of sorts, but it's just weak and has a very short lifetime.
Thank you.
Sure.
Yeah, I had a question about the hydrophobic interaction. So you said that there's an energy penalty, and does that contribute to the enthalpy term in the thermodynamic scheme? And then, because I'm assuming if you want to like have two things bring together, that decreases the entire entropy of the system. So then that's technically.
I think, oh God, I should know this better. I believe that the hydrophobic effect is almost entirely entropic. That's why it is better at low temperature than high temperature. So I think the hydrophobic effect is strongest at, well, it's a little complicated because the strength of the hydrophobic effect also depends on the strength of the hydrogen bonds. And it's it's water's kind of mission to maximize the number of hydrogen bonds it wants to make. It wants to do most of that as much as it can. Anything that disturbs that, but you're right, it screws up both enthalpy and entropy. You pay an enthalpic cost because you're making fewer hydrogen bonds, but you also pay a entropic cost because in something like this, you have disallowed a randomly moving water ever to enter in this space or even at this interface. You specifically kick them out. So I think there's contributions from both. And the other thing about the rotation, I think that the water, I think it was on the scale of times, the rotation of a water molecule is also fantastically fast. It's faster than a nanosecond to rotate. So the formation of the reorientation of water to flip around some object happens faster than the hydrogen bonds, for example, can form. Rotation is usually never a limit.
Speculated that the water might be organized on like further layers.
Yeah.
In places like the nucleus, would the cell then maybe have like suggest a preference to being ordered, like having ordered water so that if there's a disturbance in the order, it can act as like a signaling mechanism then?
I think, yeah, and you'll see later, and one of the things which we didn't get to, but that's useful to remember is that the biochemistry in the cell is so different from biochemistry in dilute solution because everything is really crowded. On the whole, the cell is 70% water, but most of that water is tied up in hydrogen bonding or moving around a hydrophobic surface. And in the nucleus, it's even worse. Nucleus is just packed full of stuff. And so there's this one useful thing to keep in mind that if you take an average protein, say, and ask in the cytoplasm and even worse in the nucleus, and ask how far is that molecule from its next closest big molecule, a nucleic acid, a protein or something, it's never more than its own body length away from colliding with another protein. So things so it's better to think of the crowding in a cell as though you are, in a gigantic line leaving an an arena or something, but you're not actually, you're not touching each other shoulder to shoulder, but that's about 50%, volume fraction people. If you don't actually pack together. And that's about how crowded the cell is. So the water organization is a useful thing because if an average thing in the cell is like 5 nanometers and it's only 4 nanometers away from its neighbor, in that 4 nanometers, there's only a couple hundred waters. And same number of, and there's also a bunch of sugars and there's a bunch of, other stuff in there. So it's not clear that you can think of this thing as just being surrounded by water the way you would if you just did biochemistry in a dilute solution. So those things change the kinetics, they change all kinds of stuff. But the cell is and this is I think that's a colorized EM or super resolution picture of the interface between the nucleus and the edge of the cell. So here's an adherence junction between 2 cells. These are the lipid bilayers. Here's some intermediate filaments or actin filaments. Here's the stuff in the nucleus. Here's the nuclear pore complex. And this is what the actual cell looks like inside. It's just so packed full of stuff. It's hard to know how to relate dilute solution biochemistry to this environment. So it's often not a surprise that what we know about polymerization rates or protein interactions what we know about that from pure systems may not be the same as what happens in the cell. But for some reason, yeah, I find this image is very kind of useful for your intuition, just how packed everything is.
Hey, Paul, this is Ram. Can you hear me from Zoom?
Yes,
Yeah, I was listening to most of your lecture. I think I missed just the first 5 minutes. I wanted to say if there are no more questions in the room. Recently, last year, we had, as part of our qualifying exams, one of the papers was about giant viruses. So there's a whole bunch of these giant viruses in the ocean, particularly. They are like micron size. And there are things that are called mimi viruses. So you can take a look at that. That was blew my mind, but I think they've been known or described at least for some time, but not discussed widely, at least in cell biology.
oh, that's very cool.